Hi, hello, welcome back to a Physionic deep dive, detailed analysis of a study. Today's episode is going to be one where I really nerd out because we're going to get into some nitty gritty mechanisms of how saturated fat affects our pancreatic health. So, specifically, our pancreas is an organ that allows for the generation of insulin. So if you've been with Physionic for a while, you know that I have a series that is already released looking at the effect that saturated fat, specifically palmitate, has on muscle insulin sensitivity. Well, we covered a number of different studies on that topic, and certainly it's something I'll revisit in the future as well. But for now, I've wanted to focus my attention now on a new series looking at how saturated fats affect our pancreas. So the flip side, so not how it affects the back end, so where insulin sensitivity on muscle, but the effect that these insulin producing cells, how those are affected by saturated fats. Now, the limitation of some of the studies that I've looked at so far, one of the limitations has been the fact that it's been only in palmitate, which is while it is the most dominant saturated fat, it's been brought up a few times. A number of people have asked me if this also applies to other saturated fats, like stearate, for example. Well, in this video, I'm going to show you some evidence that looks not only at palmitate, but also looks at stearate, as well as looks at other unsaturated fats. So we've been focused on oleate, for example, but we're also going to be looking at other unsaturated fats. So we're going to be looking at a series of different fats. We're going to be investigating glucolipotoxicity, which is something I'll explain in just a little bit, as well as how metformin could impact these results that we're going to be going into, and much more as well. So it's going to be an involved one. It's going to be a long one. If you want to skip all this and jump to the conclusions, feel free, but certainly you won't get, you'll miss about 90% of the information. And I will, of course, have a short version of this video releasing as well. So if that's released, then you can click on that and skip all the details. But if you are a nerd like me and want to actually get into the nitty gritty, then this is the video for you. Definitely buckle in, grab a snack, grab a drink, whatever you have to do. It's going to be a while as we delve through this. So if you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine. I hold my master's in exercise physiology. I've been doing cell biology research for 8 years now, coming on close to 9 years now. And that's essentially all my credentials for the time being. And let's actually look at some data. Let's analyze some data. So the question that we're going to be examining, as I mentioned, is how do several unsaturated and saturated fats affect insulin-producing cells? And those are the pancreatic cells that I was mentioning. I'm going to give you some background on all that in just a little bit. So big picture, though, what are the topics that we're going to be covering? Glucolipotoxicity, the hypothesis behind that, mechanisms of pancreatic damage, and metformin's role, as I alluded to. This information is going to be coming from a really detailed study called Saturated Fatty Acids Synergize with Elevated Glucose to Cause Pancreatic Beta Cell Death. Again, as always, I always warn that we shouldn't just base our conclusions or just look at the title or even the abstract and say, okay, well, there we go. The researchers think this, so therefore we should just, we don't even have to look at the literature. No, we have to look at the data, and that's what we're going to be doing. So to give you a bit of background on how they went about this study design, they had two different sets of cells that they ended up doing a series of experiments on. So most of the experiments are on these INS 83213 cells. So these are a rat line, so what are called immortal cells, meaning they're cells that can continuously be used over and over and over again. They just kind of replicate themselves. So this is going to be a model. And then we have another cell line that are human beta cells. So these are also beta cells, but they're from rats. And these are human, obviously taken from human donors. So these would be individuals that have already passed away and they've donated their body to science, for example. And then we take we end up scavenging and taking certain parts of their body, in this situation their pancreatic cells, and then isolating beta cells specifically, because beta cells are the ones that produce insulin, and I'll have an image to show exactly what that is. So the researchers end up incubating or surrounding the cells in glucose, so a kind of a mimetic for blood sugar, and then the varieties of fats that I'll go into in just a little bit. And then over time, after X amount of time, 24 hours, something like that, then they take measurements of the series of different measurements that we're going to be going into. So it's going to be, like I said, pretty involved. Now, there's always a critique. I always mention this because when using animal cells, so one, obviously cell culture, we have to contextualize things. So this is in cells on a dish. So this is great for mechanistic research, which we're going to be looking at a lot of mechanistic research, but the applicability, big picture, is it's certainly applicable, but we have to temper our expectations, and I'm going to do that during the conclusion, so stick around for that. Also, this is some of the cells are obviously from animal cells. So I have a video where I compare animal cell culture work to human cell culture work, looking at insulin sensitivity. So if you're interested in the comparison between the two, essentially showing, is there actual comparability? Can we base any conclusions from animal research on humans? Well, I would recommend that video because I break that down in showing that there's significant overlap between the two. Not always perfect, and again, something that needs to be contextualized, but if you can do that, and if you can get more research, which I will certainly be showing you more research than just this one study to make definitive conclusions, then you can get some great data from animal research that you can never get from human research. All right, so those disclaimers out of the way, let me go ahead and give you a bit of background here. So the way that your pancreas works is that, so you have a pancreas, and when you consume carbohydrates, typically carbohydrates, you see this sudden increase in blood sugar levels. So those blood sugar levels will increase, and that will be sensed by the pancreas. So this is blood sugar, blood glucose, which will go to the pancreas. It will be taken up by this region of cells known as the islets, and these islets have particular cells known as beta cells. They also have alpha cells and other types of cells, but the ones that we're concerned with are beta cells. So these beta cells then secrete insulin, and then that insulin then binds to all the peripheral tissues, if you think about muscles, kidneys, whatever it might be, and allows for this blood sugar that didn't interact with the pancreas, the rest of the blood sugar that has now been elevated in the blood in the bloodstream, will then be shunted into all the peripheral cells. So the insulin is, people love to use the one analogy of, it's like a key to the lock. So it opens up all the peripheral cells to accept glucose, therefore reducing blood sugar back down to normal levels. So that's how it would normally work. Now, our question is, what happens when we combine this blood sugar with the addition of fats, specifically saturated fats, which is this linear fat, or this kinked fat, which is unsaturated fat? And this is where this kind of glucolipotoxicity hypothesis comes from, in that researchers believe that if you combine sugars, carbohydrates in general, plus fats, that you can stress your cells, stress your system. There's going to be a lot of context to apply to that, and I don't want to get too far off track, but the bottom line is let's just limit our scope to understanding that if you have elevated fats in your bloodstream plus elevated sugars in your bloodstream, this would be the lipotoxicity theory. So how would those affect the pancreas, for example? And that's what we're going to be looking at. Okay, so that said, let's go into the actual data. I'm going to break this down for you so that it's going to be as easily understood as possible. If you have divergent ways of interpreting some of this data, I would be really interested in hearing that. If you have, but please have some reasoning behind it, not just, sometimes I get comments from people that just say, that's BS, man. I'm like, okay, like I need reason. Like I can't just I don't change my opinion based on that's BS. So if you have thoughts, please post them. I'd love to hear them. But again, reasons. Okay, so the first piece of data is to look at cell death. So the amount, which is pretty self-explanatory, how much cell death is occurring. And we have a physiological glucose level. So G5 is this line right here, which is physiological glucose. So 5 millimolar, which is just under what you would get for pre-diabetic individuals. So it is in the healthy range, in the quote unquote healthy range. G20 is 20 millimolar. I think that's like 400 milligrams, something like that per deciliter. So G5 is under 100 milligrams per deciliter. G20 is, I guess, yeah, it would be around that 400 milligram per deciliter range, which is astronaut. That's really high. If you have that high of glucose, you definitely have diabetes. And that's kind of raging glucose levels, which is going to be important as we look through this research. So that's what happens when. So they're looking at cell death, they're looking at these two different glucose conditions, and then they're titrating or adding slowly different concentrations of the saturated fat palmitate. Palmitate is the one that I've been focused on the most of late, but this is what they've been focused on here for this first figure. And what we find is that as they titrate more palmitate, you see that this curve starts to go upward. So at higher levels of palmitate, specifically in the high glucose condition, you see increases in cell death, really substantial increases in cell death. By comparison to if you add palmitate plus the normal glucose levels, you do not see that cell death. Now, interestingly, this is divergent from some of the other data that we've looked at from other studies that have shown cell death even at normal glucose concentrations. However, those studies used like 5.5 millimolar. So that might be just a little bit more. And maybe it could be that we're just at that cusp of like, if you go slightly above physiological levels to maybe pre-diabetic levels, you start to see those detrimental effects. But that's pure speculation. So, but off of this data, we see increases in cell death with the high glucose plus high palmitate addition. And I've got this schematic here of a beta cell being undergoing cell death. So this blebbing that occurs is known as apoptosis. And I'll get more into that in probably the next slide. Okay, so this bottom piece is to look at different fats. So I promised that we wouldn't just be looking at palmitate, although it's certainly focused on palmitate. We're also going to be looking at some other fats. So we've got our control condition, which is the condition where no fats are added. So we just have the glucose. So we have glucose concentrations of G5, 5 millimolar, 11 millimolar, and 20 millimolar. So 11 is still pathological. It's above what you would normally see. And 20, of course, as I mentioned, is astronomically high. So regardless, if no fats are added, even with these super high glucose concentrations, we don't see elevated cell death, which is what's indicated by this white box for control. On the other hand, we see this stepwise increase again with palmitate. Of course, that's what we found here as well, that the higher the glucose concentration, the more of this cell death occurs. So more of this pancreatic cell death occurs. Now, on the other hand, oleate, which is a monounsaturated fat, we do not see that stepwise increase. So this implicates specifically this saturated fat, palmitate, in having this detrimental effect. Now, on the other hand, another unsaturated fat is linoleate, which is an omega-6 fatty acid, which some people sometimes ask me about. And here we see that even though you see a slight increase here, this is still statistically not significantly increased. So therefore, we can't say that there's an increase in cell death with this linoleate condition. So the only condition where we see increased beta cell, pancreatic cell death is with the palmitate plus the higher glucose. All right, so to get a little more involved in here, we're going to be looking at two different types of cell death. So there's cell death that occurs, just general cell death, but that can be broken up into like 6 or 7 different types of cell death. We're going to be looking at the 2 major ones. Okay, so first off, again, we've got our glucose conditions, our regular plus slightly or pretty, decently high, and then really high. And then we're looking at apoptosis, which is an image here, which is considered programmed cell death. So the cell undergoes the appropriate mechanisms to destroy itself. So it's limits the amount of damage that occurs. So you have less of an inflammatory response as a result of this programmed cell death. So the cell does all the necessary cellular biology programming and initiates all those programs to efficiently destroy itself for the good of the entire body. That's one way of looking at it. And you have this initial blebbing, so it will start to segment itself so that it can be consumed by an immune cell, and therefore you don't have all these components that are going to leak out of the cell, which if those components leak out of the cell, that's a trigger for immune cells to come in a far more aggressive manner. And I'm really simplifying this, but apoptosis is, like I said, just keep in mind, programmed cell death. It's meant to happen. If the cell has to die, it's meant to happen this way. Now, on the other hand, we've got necrosis, which is kind of this unprogrammed, uncontrolled cell death, where the cell is just falling apart and it can't go through its necessary programming. So you've got these different components of the cell that are just leaking out of the cell, which would lead to mass inflammation if that's the case. So let's look at that. Do these fats have an, push? We saw that there's an increase in cell death, but do we see it be more programmed, or do we see it be more non-programmed or chaotic? All right, so apoptosis again. Okay, let me walk you through here. So they've added a fat here. So we've got our control, which is the no fat condition with just the glucose. Then with minimal fats, just to sustain the cells. Then our palmitate condition. Now here's where they added this other saturated fat, stearate, which is another saturated fat, somewhat similar to palmitate. And it's also a very common saturated fat. Not as common as palmitate, but still common enough. Then we've got our oleate, and then our linoleate. And here we see, again, with the normal glucose levels, while, again, you see these bars are starting to increase, technically there's no statistically significant increase in apoptosis, so cell death, this particular type of cell death. However, as you increase the glucose concentration, suddenly it's not only palmitate, but also stearate that lead to increases in apoptosis, so this type of cell death. And then you really see that at the extremely high glucose concentrations. But on top of that, we also see that this omega-6 fatty acid, this linoleate, also increases cell death. So there's a threshold where somewhere between G11, so 11 millimolar glucose, and 20 millimolar glucose, that it's not just a saturated fat effect, it's also a polyunsaturated fat effect, specifically for omega-6s. But monounsaturated fats seem to stay completely consistently low, so no dramatic increases in cell death, regardless of the glucose concentration. I think this piece of data alone is really, really interesting. So now let's look at necrosis, so this uncontrolled cell death. I can just go ahead and tell you all the way across, there's no increase in necrosis. So every single time these fats induce this cell death or these beta cell death, it is mediated through this programmed style. So as far as cell death goes, not that you want your cells to die, specifically, when you're talking about pancreatic cells, healthy pancreatic cells, it's at least going through the programmed method, so a programmed system. Now, to look at another measure of cell death is to look at a tunnel assay. So this tunnel assay, this tunnel experiment, essentially identifies in the nucleus. So your cells have a nucleus where your DNA is kept, your genes are kept, and within that nucleus, if you have tons of DNA damage, so a bunch of shearing that occurs, a ton of cutting up of that DNA, then you have a bunch of breaks in that DNA, and this tunnel assay picks that up. So you're able to get a fluorescent signal so that the more fluorescence that you get, you can quantify that, and that would identify then more DNA damage. And what we find is, again, you see some DNA damage, but a basal level at and you're always going to have some DNA damage, because within your nucleus you have enzymes and stuff that are constantly repairing your DNA. So it's not like it's always going to be zero. You can increasing and decrease it based on your environment and behavior and things like that. But overall, you're still going to have a little bit that occurs. Anyway, I digress. So G5, this regular glucose concentration, we see a little bit. Then with palmitate, we don't see enough to see a statistically significant increase. Then even with the really high glucose concentration, we still don't see increases in this DNA damage. And this is compared to the G5 by comparison. So, but then when we combine the really high glucose concentration, this time they've moved it up to 25 millimolar, plus the addition of palmitate, we see a statistically significant increase in DNA damage. Now, it's possible that in this situation with the low glucose or normal glucose plus palmitate, that the assay just wasn't sensitive enough, or the statistics weren't sensitive enough to pick up this difference. Because sometimes you look at the graphs and you think, well, surely there must be a difference, but to be intellectually honest, we have to say that there is no difference, and there's only a difference with palmitate plus high glucose, which again kind of feeds into this lipotoxicity mentality or hypothesis. Okay, so here, as I said, I was going to be nerding out. This is a pretty cool experiment in that I had to look this up, and I had to look up how exactly they went about doing this, but it's a really easy thing to do. So again, we've got our palmitate, we've got our stearate, both of those are saturated fats, we've got our oleate and linoleate, and this is in the low blood sugar or glucose condition and our high glucose condition. And again, of course, we see, well, I guess I should explain what we're actually looking at here. So this is a DevDace activity assay. What does that mean? Well, let me take a half a step back and explain that when your cells undergo programmed cell death, they typically activate these proteins known as caspases. These caspases, what they do is they go around to all the functional proteins that are found within your cells, and they essentially cleave them. They cut them up so that they're non-functional anymore. So they destroy the proteins by cutting them up in multiple different locations. So high caspase activity would mean that you have a lot of damage that's being done to the cells. So that's what they're measuring here. But how exactly are they measuring that? Well, and you may not really care about this, but for any nerds out there that are interested, this is how they're measuring it. So what they've done is that they've made sure that the cells express a particular protein, so it can be just any protein, that has a caspase signal. So anytime the caspases are active, because they're not always active, but anytime they're active, they'll go around and they'll cut exactly at this particular site where I've got this shown in green versus blue. And when they cut at that site, it activates this separation of the main body of the protein versus the actual green part of the protein. And this green part of the protein is a GFP, so it's a tag. It's a green fluorescent protein. So when we shine light on the cells, that green tag will then fluoresce. So we can tell when caspases are active in that once they cut that green fluorescent protein off of the main body of the protein, it will move from the cell membrane. I should have shown you some images, but the cell membrane has this outline of these green fluorescent proteins. And once the caspases go to those proteins and cut them off of the cell membrane, they'll flood into the cytosol, or the main body of the cell, which then makes the entire cell fluoresce green. Well, in this situation, green, but you can also change the fluorescence. So that's how they're able to tell if caspases are more active. And what they find is that with the palmitate, you see increases in this green fluorescent protein, which would indicate increased caspase activity. And with the stearate, again, the same thing. But again, with both of the other unsaturated fats, we do not see this effect. So this all falls in line with the previous data looking at cell death with apoptosis. Okay, so now looking back at cell death, so this is the exact same experiment. So looking at cell death, just like we did in the very, very first piece of data right here and here as well, but it's displayed like this. And in this situation now, they're adding a inhibitor of apoptosis. I believe that's right. Let me quick double check that. Okay, it's a caspase inhibitor to be specific. So it's an inhibitor of these proteins right here. So remember, this is a programmed system, meaning that it belongs to the apoptosis program, or typically attributed to apoptosis, this programmed normalized cell death. So now they're using this ZVAD FMK drug or some sort of substance molecule which will inhibit the caspases. And now they want to know, well, what happens? What if we inhibit the caspases? Do we then see a shift from apoptosis, so programmed cell death, to this unruly, hectic, uncontrolled, unprogrammed cell death, which is through necrosis that I mentioned earlier? So in the control, so without palmitate added to the cells, we see that there's no appreciable increase in apoptosis, nor an appreciable increase in necrosis. However, when we add palmitate and we use both of these inhibitors, we see that first, without the inhibitor, so at G20, so without the inhibitor, so you don't see the plus ZVAD FMK, then that means you see this substantial increase in cell death. But that's not news to us because we already saw that. So they're just repeating the experiment and they're showing, hey, we still see this increase in cell death, general cell death that occurs. Well, in this case, apoptosis, that's what this A stands for, and the N is for necrosis. So, and we see a dramatic decrease in apoptosis when we add this caspase inhibitor. So the apoptosis is largely mediated through the caspases going around and destroying all the proteins. However, when we do that, we see a slight increase also in necrosis, but we see a really dramatic increase in necrosis when we inhibit those caspases. So that means that it's caspase mediated that this whole process occurs, and that palmitate has this detrimental effect on the beta cells. Okay, so now we're revisiting the same experiments, but they're adding another element to this, which this is probably the coolest, I think, experiment of the entire paper. So here they're looking at cell death generally, necrosis, apoptosis, doesn't matter. If the cells are dying, they're incorporated here. So same thing that we're already seeing with the addition of palmitate. So these two conditions, if they have a minus, means that there's no addition of palmitate. And if there's a plus, that naturally means there's an increase in palmitate, or they added palmitate to these conditions. So you can tell that they're honing in on palmitate now. And here, this is the DevDace activity assay, which is, again, this measure of caspases, so as I described earlier. Now, what they're doing here is they're adding another drug known as etomoxir. That's a difficult one to say. I've actually used this drug, not on myself, but on the cells that I use in the lab. And what it does is, let's just separate that for a second, and let's just look at what a fat would do. So if a fat enters the cell, a hepatic cell, doesn't matter, it can go into a pancreatic cell, it doesn't matter what type of cell, but it enters the cell. And from there, largely doesn't matter what cell it is. If it's a red blood cell, it's not going to do this. But from there, once it's entered the cell, it can enter the mitochondrion. So the mitochondria, the powerhouse of the cell. Well, in this situation, we are going to be looking at it as the powerhouse of the cell, because fats can enter the mitochondrion through these three different proteins. These three different proteins have particular functions in that they change fats particular ways to allow them to enter the the inside of the mitochondria. Once they're inside the mitochondria, they can be oxidized, meaning that they can be cut up and can be used for the generation of cellular energy through ATP, largely ATP. So here we've got our saturated fat, and it'll interact with this first protein known as CPT1, or carnitine palmitoyltransferase 1. And that protein, CPT1, will conjugate another molecule known as carnitine to the fat, so this saturated fat in this situation. Then this will allow the saturated fat to enter the kind of the space between the outer membrane of the mitochondrion and the inner membrane of the mitochondrion, which will then allow it to move through what's another protein known as the translocase, which allows fats to go from the outside to the inside of the mitochondrion. And then they will interact with another protein, which is like the brother or the sister of, or the sibling of CPT1, known as CPT2. So carnitine palmitoyltransferase 2. And then from there, it will deconjugate, separate this carnitine molecule from the palmitate, or from the saturated fat, or from the fat in general. And then once it's been deconjugated, or it's been removed from that carnitine, it will then be able to go through a process known as beta oxidation, which leads to the fat being cut up and used for energy. Okay, so why did I tell you all that? The reason why I told you that, partly because I'm nerding out, but secondly, for more important reasons, is that this drug, etomoxir, inhibits this CPT1 enzyme. So it inhibits this protein right here. So now the fats are no longer able to move because they're not conjugated to that carnitine. They need that carnitine to move through the membrane, at least these types of fats. So that means that now the fats can no longer be oxidized. They just have to stay out here in the cell. So it's poor representation because you don't actually see the cell, but they would stay in the cytosol around the nucleus, but outside of the mitochondrion. And here we see that if we inhibit this CPT1, meaning again that the fats can't enter the mitochondrion, which means that they would naturally accumulate within the cell, that we see this massive increase in cell death. But not only, so it's predictable that you would see this increase with the high glucose condition, because we've seen it multiple times now. But not only that, we also see an increase when in the regular glucose condition. So if the fats can't enter the mitochondrion, that means that you get this increase in an even greater increase in damage or cell death. And also, of course, in damage, because the caspases are also highly active, not only in the high glucose condition, but also in the normal glucose condition, which means that this whole process relies at least partly on the mitochondrion functioning. So what I think is happening here, if you'll allow me real quick, what I think is happening here is that the cells are taking saturated fats, and even when they're in abundant levels and with glucose, so they have high amounts of both nutrients, and the cells are trying to burn through these nutrients as fast as they possibly can to avoid some of the problems that we're seeing here. So when the researchers block the ability to burn off the fats within the cells, the fats start to just accumulate, which leads to a series of different problems like increases in caspases. So the mitochondria are acting as sinks for these fats. And once you block that sink, then it just starts to pile up within the cells and leading to worse and worse effects. Hopefully that makes some sense. Finally, we're going to be looking at metformin. So this is just a quick piece of data looking at metformin. If you're not familiar with what metformin is, it's been considered a wonder drug, a miracle drug, and I've had a few people push back and say that there's evidence against that it causes Alzheimer's and stuff. But admittedly, I looked at some of that data, and I didn't run across that many studies. Most of the studies that I ran across said that there's no connection, or that there's a weak connection, or something along those lines. This was a while back that I looked at it. So whatever you feel about metformin, it's helped a lot of people with diabetes, that's for sure. It has had other positive effects. Are there potential negative effects? Yeah, maybe. I don't know. I haven't looked into it. So, but this is not about like some glorification of metformin. The point is that now we're going to be looking at how metformin, which is a ubiquitous drug that's often used for a series of different ailments, has what that has in terms of an effect on these cell death measures that we've been looking at. So what does metformin do? Well, metformin specifically doesn't actually activate this protein as AMPK. Let me take a step back because I'm getting ahead of myself. What is AMPK, if you're not familiar? I always assume that people have been following Physionic for a while, which is not the right way to teach. AMPK is a master regulating protein that senses the energy state of the cell. So if you have high ATP levels, so high cellular energy levels, relative to AMP levels, which are, or ADP levels, which are lower energy state levels. So high ATP to low AMP would mean a high energy state of the cell. Low ATP or high AMP levels or high ADP levels would indicate a low energy state of the cell. In a high energy state of the cell, high ATP or low AMP or low ADP, AMPK is not active. However, in the reverse, when you have lower ATP or higher AMP, then AMPK is more active. Now, metformin affects your mitochondria in such a way that it reduces ATP production and therefore activates AMPK through this indirect mechanism. ICAR is another drug. It's an AMP mimetic, meaning that it mimics the molecule AMP, which I said, if you increase AMP, you activate AMPK. So what they're doing is they're adding ICAR or they're adding metformin, and both of those have effects on AMPK. One of them is more direct, ICAR, and the other one is more indirect, metformin. And they're seeing what effect that has on cell death. And there's a little bit of wishy-washiness to this, but we can actually get some results here. So with palmitate, again, plus means that they've added palmitate, minus means that they haven't added that particular drug. So with metformin, they see that there's no change compared to when they don't have palmitate and both metformin. Okay, so that's not a shock. But when they add palmitate, of course, we see the increases in cell death. We've seen this like 4 or 5 times now. But when they add metformin plus palmitate, we see that there's a dramatic reduction in cell death. If we look at ICAR, we see the exact same effects. Without ICAR, there's a huge increase in cell death, and then with ICAR, you see this dramatic reduction. So where the wishy-washiness comes in is, what is the effect? What is AMPK doing that's protecting these cells in this situation? Maybe it increases mitochondria so that there's more of a sink for palmitate. That's a possibility. Maybe it's quieting some of the cell stress molecules that would normally go around and communicate to the rest of the cell. We are under attack. We are highly stressed. Maybe it's affecting cell signaling, because I said it's a master regulator of the cell, of cell energy, but it has wide-reaching effects to all kinds of. It's like the opposite of mTOR, if you've ever heard of mammalian target of rapamycin. So it's a different protein. They're both antithetic to one another, but they work really closely with one another. And AMPK has wide-reaching effects all over the place beyond just cellular energy state. So it's possible that AMPK may quiet down some of those stress proteins, for example. I'm really speculating. I don't know, because the data doesn't show anything in terms of how this effect is happening. But what we do know is that metformin helps and ICAR helps. And I'll bet you that if you threw an exercise which also activates AMPK, you would also see this effect. To what degree, for how long you would see this effect, I don't know, but there's certainly a possibility. But that's speculation on my part. Okay, so that said, all of that said so far, elevated glucose, in conclusion, if you've jumped to this, you have missed so much information. But elevated glucose plus saturated fat, in this case, I can finally say it's more than just one saturated fat. It's definitely at least two, the two most common ones, palmitate and stearate. Elevated glucose, so high glucose levels in pathological states, plus saturated fat induce pancreatic cell stress through caspases, which we definitely saw a lot of data for, ceramides, which I skipped over that data, but that is also shown in this paper as well as some of the other papers that we've looked at, plus DNA fragmentation leading to DNA damage. These effects do not occur largely with unsaturated fats, except for linoleate. We saw with super high glucose concentrations, I think we saw increases in cell death. So, but in all the other measures like caspases, DNA fragmentation, things like that, linoleate did not seem to have negative effects. So what does that all mean? Again, it's cell culture. So am I going to sit here and say, well, now we have definitive proof that palmitate or saturated fats have these negative effects on pancreatic tissue? No, you can't say that. Until we have human trials, more human data beyond, I didn't even point it out, but some of that data was in human cells. So until we have actual human trials that we can actually do some more rigorous experimentation, we can't say that. But we have looked at other human data, some actual human studies, and for the most part, they've indicated up to this point, whenever this releases, up to this point, the studies that we've looked at have shown that, yeah, saturated fat decreases insulin sensitivity. It seems to also affect insulin secretion. So this isn't like an isolated paper. And it's not just cell culture that shows some of this stuff, although there's certainly limitations in human data because we can't, biopsy humans like we can here. So I think cautiously we can say that this adds evidence that saturated fat has more negative effects, has additional negative effects when you pair it with high blood sugar levels. I'm not saying that if you consume carbohydrates plus fats for one meal that suddenly you're going to be damaging your pancreas. What I'm saying is that repeated exposure to continuously eating carbohydrates plus saturated fats over and over and over again over years could have some detrimental effects on your pancreas. But we would, of course, need more data on that. And keep in mind that this is probably more applicable to people with diabetes. But that said, some of the data that we looked at, it's also possible that the research didn't have enough power, meaning that there weren't enough tests done, there weren't enough samples taken to be able to figure out some of the differences even under regular glucose conditions. That's also a possibility. But based on the data that we have so far in this paper, we can only say that high blood sugar levels plus the consumption of fats, only saturated fats, leads to glucotoxicity, leading to stress and damage of pancreatic cells. That is not the case with high glucose plus unsaturated fat. So if you have diabetes, for example, then it might indicate that with your high blood sugar levels, at the very least, you should focus on unsaturated fats if you have to be eating, copious amounts of fats, because that tends to lead, especially monounsaturated fats, that leads to improvements in, or at least no added decrements or no added damage to the pancreas. There's so much here to unpack. It's difficult for me to just distill this all into just a few sentences and to make this highly applicable. But ultimately, more and more and more data that we're going over is showing that saturated fats just don't have the greatest effects. I mean, at the very least, we can say saturated fat doesn't help. It does not help. I mean, I think I can pretty confidently say that up to now that we've covered like 8 or 10 different studies on the topic. Okay, I'll get off my soapbox, and if future research comes out that maybe adds some context that I had never considered or that we had never considered, then I'd be more than happy to change this thought process and amend how the context that we can apply here. So with that, I hope that this was informative, at least fascinating, to learn a little bit more about how cells work and how intricate the different systems are, how they work together so well, and a little bit on glucose and insulin and fats and all that stuff. And I'll hope to speak with you in the near future. Have a good one. Bye.